Welcome, everyone. All right, we're going to go ahead and get started. So welcome to our first webinar for Phenology Week. My name is Erin Postumus. I'm the Partnerships and Projects Manager here at the USA National Phenology Network. We're so glad that you are celebrating Phenology Week with us. Our intention this week is to celebrate all things phenology and acknowledge all of the wonderful contributions of our Nature's Notebook observers, our local phenology programs, and all of our various partners. So if you are not familiar with phenology, you're going to be learning all about it today. I did want to point you to a couple other things that are coming up this week if you're not yet aware. We have a few more webinars this week. So we've got a phenology wheel workshop tomorrow where you can actually take part in putting together your own phenology wheel. And we'll point you to how you can use Nature's Notebook data to help construct that. We also have a keynote presentation from author Mark Heinlein coming up on Wednesday. He's going to have some great stories about phenology to share with you all. We also have a update on our Nature's Notebook mobile app. I know everyone's very excited to hear updates on that, so hope you will join us for that on Thursday. And then Friday, we have a researcher Q&A with 3 researchers who have used Nature's Notebook data recently. So we'd love for you to join that, and you'll have the opportunity to ask them questions. So while we're getting started here, I do invite you to put in the chat. We'd love to hear where you're calling in from. Also, if you want to share anything that's going on with phenology right now, maybe you're seeing signs of spring already where you are, but feel free to put in the chat where you're calling in from and a little bit of something about what's going on, maybe something that's blooming right now or something that you're seeing. So we are going to be having a presentation today from Dr. Theresa Crimmins, who is the director of the USA National Phenology Network. Theresa has been with the network since 2007, and we're really fortunate that she's going to be walking us through a little bit of background on Nature's Notebook and local phenology programs and telling us about some really neat data that are being collected as part of Nature's Notebook. Really fortunate that Theresa is kicking us off today with the first webinar for Phenology Week. So we're going to kick it over to her. So before I have her share her screen, I did want to launch a quick poll. We want to try to get to know a little bit about who's joining us today in terms of your experience with Nature's Notebook. So let me just get that going here. All right. I'm going to launch this poll. So we're just interested in answering whatever you feel like best fits you. Maybe you're a longtime Nature's Notebook observer. Maybe you're a new Nature's Notebook observer who's just joined us recently. We might have some local phenology leaders here on the call today, Local Phenology Program participants, or maybe you're just here to learn. All right, we've got a bunch of people that have answered. I'll give it a little bit more time. It looks like we have people joining us from all across the country. So it's like some places are still seeing snow, but there's a lot of activity happening in other locations too. Lots of spring happening in the Southwest and some wacky winter in some parts of the country. All right, I'm going to go ahead and end the poll and share the results. So it looks like we have a bunch of new folks on the call who are here to learn, so that's wonderful. We also have quite a few longtime Nature's Notebook observers, a few new folks, a couple local phenology leaders, and program participants as well. So a nice mix of everyone here today. All right. So with that, I am going to go ahead and pass it over to Theresa.
Thank you so much, Erin. Did we share? I'm sorry if I'm messing up the poll. I am a co-host here and sometimes things don't go well. Okay, you can see my screen, right?
Yep, we can see
Great. Okay, thank you so much for that great introduction. And great, thank you all so much for being here this morning. It is really great to be able to celebrate spring and so fun to see how many folks are here excited to share. So we've reached the start of climatological spring. Climatologists define seasons just using 3-month moving windows, and climatological spring begins on March 1st, so we're past that. We're just about to astronomical spring, which is defined by the spring equinox, which this year is actually on March, I think 20th. So a few more days on that. And then the third way that we usually celebrate spring here at the Phenology Network, and I imagine most other folks do, is when we start to see those biological moments, those things that seem to indicate like, okay, we are transitioning out of the long, cold, dark winter, or whatever your winter looks like. We don't actually have long, cold, dark, lack of green here in Southern Arizona, but we do see a lot of things that scream spring to us, and we're starting to see that as evidenced in your comments. So this is going to probably do a pretty good job at reflecting what a lot of you have been reporting across the country, and thanks very much for sharing. In case you're not familiar with this, one of the things that we do here at the National Phenology Network is offer a series of real-time maps that indicate where we're at, either with relative to particular events occurring, or the map that's really the most popular at this time of year is whether locations across the country have reached the conditions that are associated with the onset of biological activity yet. And so the map that we have that gets circulated the most often is this one, which is actually an anomaly map, meaning it shows where locations not only have reached the conditions associated with the start of spring, but also whether they have been reached earlier or later than average so far this year. And when we say earlier or later, we're comparing this year to the last 3 full decades, so 1991 through 2020. And so this map was as of yesterday. And so any locations that are shaded in a red tone on this map are basically seeing the start of spring earlier than average this year. And the darker red it is, the earlier it is. And some of those locations up in Montana and South Dakota and Nebraska are over a month early this year. They just really have had very, very warm conditions this spring. And then the locations that are in the blue tones are actually seeing the start of spring arriving later than average this year. And so interestingly, it's not that much of the country, but it is the East Coast. And that is what tends to shape the national news cycle. And so interestingly, we haven't had a whole lot of discussion about whether spring is early or not in the national news media as of yet. We have this map, we call it our leaf index map. You can look down here on the lower left. It's a bit of a misnomer. The name derives from the fact that this model, the conditions that are used to create this map are based on when early season shrubs like lilacs and honeysuckles leaf out, but it doesn't necessarily indicate when everything is leafing out. So I like to think of it more as just what's the very beginning of biological activity in spring. And that includes both plants and insects and birds and other things that are early season as well. We have a companion map that we call the bloom index, which again is a bit of a misnomer. It's named that because it basically indicates when are you reaching the conditions that are associated with flowering in those early season shrubs like lilacs and honeysuckles. But what it really represents is more just how are we doing at kind of a few weeks in on spring, and are we achieving that early part of spring earlier than average or not? One of the events that's actually associated pretty closely with this bloom index is actually the start of leaf out in overstory canopy trees. So if I had my druthers, I would name it something else. But it's interesting to kind of toggle between these maps because what you can see is that locations like northern Texas and Oklahoma that were not super early for the very beginning of spring actually have seen a bunch of warmth rapidly since that point in time and now are really picking up steam. And so what this really reflects is the fact that we've had a lot of warmth lately in particular parts of the country. Again, not the East Coast, kind of the middle of the country. Aside from, I know there like a lot of the northern states are still buried in snow and are facing major storms right now. So again, it's a very mixed bag out there right now. And then when we look ahead, I snagged this from NOAA yesterday. Where it's been warm, it's gonna keep being warm. And then where it's been cold, it's gonna keep being cold for the next few weeks. And so we probably still won't see a whole lot of signs of spring in the upper New England states. But here in Southern Arizona, since we've had a really record-breaking warm winter and spring and a lot of rainfall, which is actually kind of anomalous for us these days, our fall, winter, spring rainfall has been really, really good. We have seen amazing wildflowers, and I've been out on a number of hikes recently, and it's been so fun to see just every color of the rainbow in flower. These are a lot of the species that are more common on the landscape in the spring when things flower, because we don't have a good bloom season every spring. And then a number of species that I don't typically see as frequently, and we've seen those too. And then also some animal activity. If you can see him, and he's hiding in the bushes here, but got buzzed at by a big rattlesnake just the other day too. The downside of all that rain and that abundant warmth that we've had lately that has caused all those annuals to flower and other plants to really take advantage of the good conditions is that we're also experiencing kind of explosive growth in invasive grasses down here right now. And that definitely can be a problem because as they dry out and it gets warmer, they can carry wildfires in ecosystems that are not wildfire adapted. So this is kind of the very sad downside. The buffalo grass is especially a nasty one here, and we're seeing some serious invasions right now of that. Okay, well, I really appreciate you filling out the poll because it's helpful for me to know that we've got kind of the whole rainbow of people present in the room today. Folks that are all not only longtime Nature's Notebook observers or even participants in groups, but folks that are here basically Nature's Notebook curious, learning about phenology and about the platform. So I'll take a moment to just explain things a little bit further. Nature's Notebook is the platform that the USA National Phenology Network offers that enables pretty much just about anybody to document what's happening seasonally in plants and animals. And what we really encourage is that if you're going to participate, you choose a location that is very accessible to you, because what we're really seeking is repeated observations frequently. When you're observing a plant, we invite you to make observations on that same individual plant through time. And if you choose to do animals, then it's animals at the same location. And then each time you go out to make an observation, you're presented with a series of questions that are asking you about the status of that individual. And so if it's a plant, you're prompted with questions about the leaves on the plant, the flower or flower buds, and then the fruits. And the most basic elements of that are they're phrased as yes/no questions, like, do you see flowers or flower buds? And you answer yes or no. Do you see open flowers? Yes or no. And then with repeated observations with answers of yes or no, that eventually constructs a time series that enables an analyst to be able to identify when was that event not occurring, when did it begin, and then also did it start and stop in the spring, did it start and stop in the summer or the fall, which sometimes can happen because of, say, drought conditions or other funky weather. And then we also, in kind of the more advanced style, we invite you to answer what we call the intensity or abundance questions. So if you say, yes, I do see open flowers, then you're prompted with a subsequent question that asks what percentage of the flower buds on the plant that you're observing are open. And there are some bins. And so it's fewer than 5%, 5 to 24%, 25 to 49%. And so again, if you're making those kinds of reports frequently and consistently on the same plant, it allows you to then construct a picture of, what was the ramp up in the presence of that particular phenophase and how was it expressed over time. Anybody can participate, and you can participate as an individual observer, or we have this other opportunity called Local Phenology Programs, where folks can share the fun or responsibility, however you choose to see it, of making repeated observations on the same individual plants or animals at a site. And we launched that functionality in around 2012. And since that time, we've had more than 600 local phenology programs contribute data at one location or another at some point or another. And last year we had over 250 such groups contributing data. And those groups usually are typically how this model is implemented is that it's a group of folks that are already organized for one reason or another, and then they adopt the activity of tracking phenology. And so oftentimes it's volunteers or docents at a nature center or an arboretum, or maybe a friends group with a wildlife refuge or a national park. But sometimes it's also a classroom. Sometimes professors get creative and set up a long-term monitoring site on campus, and then the students in a course will make observations in one semester, and then it gets rotated through. The same plants are observed one year after another, but the students that are making the observations clearly shift because it's whoever's enrolled in the class. Sometimes it's Master Gardener or Master Naturalist chapters. But what we found is that this is a really excellent model because it tends to not only result in more regular data collection, it also tends to perpetuate for longer. Groups typically do collect data for many more years than individuals do for whatever reason. Also, it seems like there's a whole lot of social benefit to it too. Group members tend to want to come together, share what they've seen. There's enthusiasm. Oftentimes the data are higher quality because the group members kind of check in with each other or maybe correct each other if they're misunderstanding a particular phenotype definition. And some groups have kept with things for a very long time and really take it far. Some groups I know for sure over the winter will sustain enthusiasm and activity by having book groups or potlucks or all sorts of really cool learning opportunities. And so it's a great model and we're really excited about it. So one of the things I'm going to do today is dig into some of the data that have been contributed by one of those groups, one local phenology program in particular, that has been in existence and very regularly participating in Albuquerque, New Mexico for over 12 years. And part of the reason for that is, for those of you that have been hanging around the National Phenology Network, we've been around since 2007, and we've been collecting data through Nature's Notebook since 2009. We've been talking a long time about how long-term data and data collected across a huge location and especially temporally rich, so regular repeated observations at the same site, are very valuable for understanding how phenology is changing, how it's tied to different conditions, and how that can be used in real world applications. But we've done a lot of hand waving about exactly how to summarize data that are being contributed, especially by groups. And we're just now really starting to have enough data at some of these sites that we can begin to really start to dig into that. And so this, I felt like, was a cool story where we could take one group's data and drill deep and see what we can tell, what kind of stories we can tell with it. And then we can use that hopefully as inspiration for how it could be applied to other groups and in other applications as well. So I was fortunate to visit this group just last month. I was in Albuquerque for a meeting to give a presentation, and I got to go visit the site and meet the folks and tour where they make their observations, and it was delightful. It was just fabulous. If you're not familiar with Albuquerque, it is a fabulous city in north central New Mexico, and this is the Rio Grande River that cuts right through the middle. And right over here, there's a golf course. That's what this green blob is. And then right kind of along the river, both kind of to the north and to the south of that golf course, is this fabulous institution run by the city, I'm pretty sure, called the Albuquerque BioPark. And there's a zoo there and an arboretum and all sorts of trails and what else? It's phenomenal. It was so fantastic. And it is also the site of some really great long-term phenology monitoring. And so here's a zoom in on the place where this group actually is collecting the observations, again, along the Rio Grande River and in this green space. This particular team, called the Albuquerque BioPark Phenology Group, I think, has been making observations on the same individual plants and plants, I think it's only plants, every Monday for 12 years. And when asked why, they said, well, nobody really knows exactly what's going to be useful in the future. And we know these are observations that are just collected at this one site, but they can be part of this huge picture that the NPN is constructing. And that is true. The observations that they are contributing are part of a record that is now more than 45 million records in scale. That's how many records have been contributed to Nature's Notebook all across the country over the entirety of our existence. And so this is kind of like one long, very, very narrowly focused geographically, but temporally rich data set that we're going to focus on right now. but it can be put into that context of the whole national scale data set as well. So here's the team, and I, again, was so fortunate. I got to meet all 6 of these wonderful folks, Marcia, Sheila, Allison, Judith, Sandy, and Carl while I was there. And they took me out and they showed me their plants, and it was just so great. And I got to see just a little bit too about exactly what is it that keeps this group active and enthusiastic, and how do they keep at it. And one of the things that I feel like is really special about this group in particular, that they came up with on their own, is that they have scheduled it such that they have 3 observers each week. They go out every Monday, 3 people. And what they do is they make sure that at least one of those folks overlaps and also goes out the next week. And so they all get to rotate through. They spend time with each other. It changes who they're getting out there with from one week to the next. And you have that consistency. So if one person, if the group of 3 goes out the first week and they say, yes, we do think that this plant has breaking leaf buds, then the next week, if the new folks coming out are like, what are these breaking leaf buds? We're not sure. The person who had been there last week can say, well, we had this conversation last week, and this is why we think that this is this. And so there is some internal consistency built in there, which is really quite clever, I think. And I will be honest, um, having things bob back and forth from one week to the next is a challenge that we have confronted. We've identified in our data set, because when you're trying to make a distinction about are we actually seeing breaking leaf buds or not, it can be really challenging, and not everybody shares the same understanding of what constitutes a yes answer. And so something, a model like this actually can do wonders for helping address that kind of inconsistency that we sometimes see in the data coming in. They have 12 species of plants that they're observing. I just put 9 on here because it looks better on the picture. But here's the sample. There are both trees and shrubs and some forbs, meaning just herbaceous plants too. And some cool things about it is that they have expanded things over the years. And so what this plot is showing is that in the orange bars, the number of individual plants that are under observation, and you can see that in some species there's just 1, and in others there's 6 or more. Some of that I learned as I was there. You have to deal with the vagaries of the institution that you're sitting at. Sometimes they run over your plants, sometimes they stop irrigating them. Things die, things just kind of disappear, even if they have good signage. So that's what is reflected in some of these. And then the number of years there too. And so some of the species have been under observation in all 12 years by the group, and some have been more recent additions. And so I share this because I'm going to go through in deeper detail results just on some of the species that have been under observation for the most years, because we have the greatest sample size there to work with when we look at years. So what we're going to do here, what we're going to walk through here is a couple of questions that are going to be specific to these data here for Albuquerque BioPark, but that are, again, inspirational for anybody, really, anyone who's observing phenology at a single site, and especially any local phenology program. And so I feel like this is us kind of pulling back the curtain a little bit here as our team, the National Phenology Network team, is trying to figure out exactly, okay, now that we've had so many groups contribute so much data for so many years, how can we share that back with you all? What are the most valuable insights? Maybe for you to just see in your data, what are the most valuable insights for you to maybe be able to take back to any management at your organization where you're monitoring? They might be able to, they might care to know this kind of stuff. And what kind of insights might actually be indicative of the kind of change or other patterns that are becoming apparent in the data that would be really valuable to understand. And so in kind of order of increasing complexity, we'll walk through basically plotting when do the things occur, which is kind of over the course of the whole year, and how consistent is that? When do the phenophases begin? Because that's what we call onsets. Onsets are usually actually what analysts are the most interested in, because if we're thinking about whether things are changing, oftentimes it's when did the thing start, and is the thing advancing earlier or shifting later? as a consequence of some changing environmental conditions, that is really the question of interest. So we kind of narrow in on onsets there. We could also look at ends. We're not going to today though, just because there's not enough time. Then the question that everybody always wants to know is the timing of something changing. Can you see in our data whether something has shifted earlier or maybe later? Because that is so often what brings people into phenology observing, is that they hear that, well, as the globe is getting warmer, we're tending to see earlier onsets, earlier starts to phenological events. And while that is largely true, it's actually extremely variable across species and across space and across season. And so it's a very fair question, and one we will start to try to poke at here. But then the thing that you really can't decouple it with is the fourth question, which is, well, why? If it's shifting earlier, is that must be because there is a relationship with another variable, a explanatory variable, and it's typically temperature, but sometimes it's in desert environments like the Southwest, oftentimes it's available moisture. And so we're going to look at both of those variables here. And so these numbers 3 and 4 really kind of go hand in hand, and we'll see that. Okay, so to get us started with the first question, when do things happen? I have a plot here for golden currant, which is a deciduous broadleaf shrub. And I'm just going to say right out of the gate, I didn't make these plots. Erin Zylstra from our team did, and she is extremely talented. So I won't take credit for work that isn't mine. She made these great plots. And what's really fantastic about them is that they show an awful lot of information in here, all compacted in at once. So what we're looking at first off is across the x-axis is the months of the year. And then what she did was take all of the observations for, I forget if they have multiple golden currants, but I think they do. So Erin took the observations for all of the different golden currant shrub individual plants in all of the years, all 12 years of observations. And then what she did was basically what we're reflecting here is the proportion of the observations in all those years for those multiple plants on any given observation date over the course of the year. And I think this is broken down by week. What was the proportion of individuals or observations that were expressing yes? And so in a situation like this, where the bubble is all the way on 0, basically how you can read that is for golden currant in this first week in March, you can pretty much guarantee that that plant is going to have leaves. Green refers to the leaves. And that means that in the first week of March, every time they looked on every plant in every year, the golden currants did have leaves. So how we can look at that is, okay, this is a really good indication of when the plant has leaves. And what we can see is that there is the increase from no leaves. It basically almost never had leaves in January. And it increases, it shows the likelihood of having leaves increasing, that basically increases over the month of February. So you're at about 50% in the beginning or in the middle of February. So it's like a 50-50 chance that the plants would have leaves in the middle of February. And then how we can continue to look at this is how long does that proportion stay high? And it stays high pretty late in the year. So again, the fact that it's not pegged at 1 means that there were at least some instances where when the observers went out and looked at the plants in April, May, June, July, August, September, there were at least a couple of situations where the shrubs did not have leaves, but most of the time they did. And then really it's in October and later where things start to significantly drop off. And by the time you get to November, you can again pretty much be guaranteed that there won't be leaves on those shrubs. We can do the same thing for the other phenophases. The flowers are in light orange, that's basically flower buds. And then the open flowers, we can see it's a fairly tight peak that occurs between March and April. And it doesn't even go to 1. So there's years where it didn't flower at all. If it were going to flower though, you can basically plan on it being late March, early April. And then there was a tiny little blip in late June. So probably one plant, one random year actually had flowers at that time of year. And then also we can look at the fruits, and the fruits are highly variable. They happen mid to late summer. but again, not super consistently. A lot of times it looks like there was just none. And that might be that, and I don't know this off the top of my head, this might be a dioecious plant, meaning that they're observing male and female. And if that's true, the male plants would never have flowers, and that would explain it. Someone can look that up. And then the colored leaves, you can see starts to come on in May, but again is really variable. So these plots are really cool for being able to kind of get a global picture of when you might expect things to occur in the different phenophases in a species, and how much you could basically bank on it in any given year or at different times. And then it gets really fun when you start doing that among multiple species, because then you can really start to see how much stuff can vary across species too. And so on the top row here, we've got Eastern Cottonwood. In the middle one, we have, it's labeled as stretchberry throughout these plots, but apparently nobody knows this plant is stretchberry. Apparently it's New Mexican privet. So whenever when I said stretchberry when I was there last month, everybody was like, "What? What are you talking about?" So New Mexico privet, a shrub, and then four-winged saltbush, another shrubby thing. And it's really fascinating, I think, to see how much things vary. So clearly, the top two are deciduous, and you can see very clear indication of that. Eastern Cottonwood starts, it does not have leaves at all until about April. Then we start to see it, and then it definitely has leaves always, all the way till about late October, and then they drop. About the same for the privet, but then four-winged saltbush is just a little more variable, and they hang on all year long. So it looks like it's dropping and gaining leaves inconsistently among individuals and among years here. That's what that's suggesting, because it's never really at zero or pegged at one, at a proportion of one other than in May. Some other kind of fun things that we can see here are that the period of colored leaves is pretty consistent across the top two species. The bottom one doesn't even bother with colored leaves. I think that's not part of the protocol for that one. The period when flowers and fruits are present does vary across the species. And flowers in particular, that matters for pollinators. Although wind pollinated plants, just kidding. Cottonwoods are wind pollinated, and so pollinators are not going to take advantage of that. And then the fruit availability, that can matter for wildlife and other things that rely on the fruit. And so these may be not the best example for that, because I don't actually know what their fruits look like and who consumes them. But this is the kind of comparison that you could construct if you wanted to see whether there was going to be pollen availability for pollinators throughout the course of the year, or if there's the potential for gaps. And same thing for fruits, for mammals and other critters that rely on them. Okay, shifting gears a little, now we can focus just on the onsets. So again, these plots are your global picture of when are all the different things happening. And I feel like they're really interesting for being able to see what's happening, what's not happening, both within a species and among a species. But then if we're really kind of interested in trying to answer those questions about what are things changing, then we've got to narrow it down usually and look at either the peak of when it occurs or the onset. And onset is what's more common in the literature, so that's what we're focusing on here. And so these plots are a transformation, basically. They present the same information as the previous plots, but it's presented in a slightly different way. Basically, what we did was focus on whenever a phenophase is what these are, that's the word we use for all these different things that we ask about on the plants. When did basically somebody transition from saying, no, 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 I'm not seeing it, to yes, I'm seeing it? That is how we define an onset, is from when we go from finally somebody not saying no to the thing to, yep, I see it, I see it. And these box and whisker plots, basically how you interpret these are, the middle bar is the median date that yes was first reported, and then they are interquartile ranges here. And then if you have dots, those are outliers. So that's just some like really, really late or actually really early relative to the data reports. And the tighter the box, that means the more consistent the onsets are among the individuals and among the years for that phenophase and species. And so the tightest one we have here for eastern cottonwood is open flowers. And so again, what that means is among all the different eastern cottonwood trees that they're observing, and I know they have multiple trees, in all 12 years, they pretty much said, yes, I start to see open flowers in that second week of April, almost without fail. There was this one funky outlier where it was the tail end of February, but beyond that, it's very tight here around about a month window. And then on the other end of the spectrum, colored leaves has a very wide window of opportunity. They have observed colored leaves as early as late May, and they have seen colored leaves begin as late as mid-October. Um, and so that's just a very variable onset. When it starts really varies. And then I should have pointed out too, the number over here, the value reflects how many different individuals and years is these bars are summarizing across. So some of them are pretty big. And so again, we can do the same thing where we compare across species, and that can be kind of fun because we can look and see, do we see the similar patterns of, tight windows or very wide windows of opportunity for different phenophases to begin among species, and are they starting at the same time or not? And not super unsurprisingly, we tend to see the flowers occurring in spring, basically, but we do see them not occurring at exactly the same time. The privet or stretchberry looks like it tends to start flowering sooner than the four-winged saltbush, pretty much without fail. There's a little overlap between those two. Sorry, that's the presence of flowers. The open flowers definitely are stretched more apart. And so this again can be a good way to look to see how consistent are things from one year to the next and among individuals. And just kind of like basically when does the thing begin? But again, the question that everybody always really wants to know are they changing? Okay, so this is where we get to the two conjoined questions because we want to look at the simple way of thinking about it is just has the timing of the event advanced or delayed over the period of years that we've been observing it. And so here, in this instance, it would be over the past 12 years, has something that the observers have been looking at gotten earlier or later. But what we know to be true is what prompts plants to either put on their leaves or put on their flowers or open their flowers is some internal cueing, but that cueing really comes from environmental conditions. It has so much to do with what's the day length, what's the recent temperature been like, what's the moisture availability, what was the winter temperatures like. And so you kind of can't really do this part in isolation. You've kind of got to look at the environmental variables. And then oftentimes, as we'll see, you might get a trend of popping in one that doesn't show up in the other, and then that can give you even greater insight as to what's going on. Okay, so let's step through this. All right, so the first more basic kind of question is something happening? Is it advancing earlier or later? And so this is kind of what that kind of plot would look like, where we've got, I should have made this more specific. Let's pretend we're talking about leaf out in the cottonwoods. So this is all made up data. So apologies if this is really nonsensical. But how I have it plotted is that the year is on the x-axis and then the day of year is on the y-axis. And when I say day of year, what I'm doing is we take the whole calendar year and just sequentially number the days. So January 1st is day 1, and December 31st is day 365 or 366 in a leap year, and July 1st is like 182, and you can just kind of guess. Like today is probably around day 100 and something. And so in this hypothetical example, our Eastern Cottonwood tree in 2011 first showed, the observers first said yes, we see leaves on day 30. And then the next year, they first said, yep, we see leaves on day 35. And then the next year, they said, they first said, yes, we see leaves, not until day 42. And then the next year, it was back at day 31. And so you could treat this as a time series if you wanted to, but effectively, it's just when did the things happen in each year? And what we can see is that in our hypothetical example, but this is fairly reflective of what we see in the real world is that there is a lot of variability from one year to the next. And so we can fit a line through it if we wanted to see if there is any actual trend there. This is fake, so I don't think, I didn't calculate an R square, an R value or a P value, but basically, if you wanted to estimate if there was a trend, you'd be looking to see, okay, this line did have a bit of a slope to it, but how much of this interannual variability did it actually seem to explain, and is it a meaningful trend or not? So those are the kind of diagnostics that we apply to interpret it. But then we can take it that step further and go to the second part where we say, okay, well, is any sort of trend that we're seeing because it is associated with an environmental variable, and that variable actually has some sort of variability from one year to the next too, and so the things are covariating. That's what we're actually trying to identify. And so this is a situation where I said, all right, well, maybe it's average winter temperature that really has the influence on when the cottonwood trees leaf out. And so let's look at that. And so again, this is made up data, but we have year on the x-axis. Now we have average winter temperature on the y-axis in probably degrees Celsius. I probably should have had. And again, we could just kind of read this as, okay, in year 2011, that average winter temperature was 10 degrees. And in the next year, it was 15 degrees. And the next year, it was 17 degrees. But then the year after that, it was back down to 12 degrees. And so again, interannual variability. And then we can take those two things and put them together. And so on this third plot, this is where we get to that, is the timing a function of that environmental condition? We now take the years are now captured in the dots themselves. And what we have is that average winter temperature, which is the variable that we think is the one that's responsible for when the thing leaves out across the x-axis, and then the day of year that the thing occurred on the y-axis. And what we begin to see here, again, in this fake data, but with a much tighter fit, is that when the average temperatures are cooler, I did this backwards, it should be there. See, I told you it was fake data. In this pretend example that makes no sense at all, when the average winter temperature is cooler, the tree is leafing out sooner, and that when we have warmer years, the tree is leafing out later. This is a lovely, nice, tight fit that is actually the opposite of what we usually see in nature. Usually it's the opposite. When we see warmer years, things come earlier, and cooler years, things come later. But hopefully it still makes sense. And then we can calculate, again, a line of fit with the diagnostics there to see if this is a significant relationship or not, which is typically how these data are analyzed. Okay, so let's do that for real with these data, with these observations from the Albuquerque BioPark. And again, I just pulled out a couple of the species with the best data. And again, this is all Aaron Zylstra's great work. So I hope I can do it justice. But basically what we're looking at here is, again, those trends over time. We're just looking to see over the period of the record that the Albuquerque BioPark folks were making the observations, do we see things drifting earlier or later? So we've got 3 different phenophases here for Eastern cottonwood in the spring. We've got leaf out on the left, flowers in the middle, open flowers on the right. In all 3 plots, the year is across the x-axis, and then, apologies for not labeling this, the day of year is on the y-axis. And so in all 3 cases, the dots, again, are the different individual plants in the different years. And we have a little bit of jitter just so you can see where, when there's multiple individuals, they're clumped together because in each year, there were multiple plants being observed. And again, oftentimes all 3 trees would first be recorded to have their leaf out on the same day of year. So that's why they're clustered together. When we look at that time period, we do see a lot of variability. When we look at the spread of the points, she did fit a line, and the shaded zone around the line is the confidence interval. And we have the slope of the line provided here. So yes, this is a positive slope, meaning it looks like leaf out is shifting later. But what we're really looking for is where the confidence interval does not cross 0. That's where it would be statistically significant. And so we don't have that with leaves. And so this is really kind of a not very meaningful relationship. And the same is true for the open flowers. But when we look at flowers, that confidence interval actually does not cross 0. And so that actually is a significant relationship. And over the period of record, we do have, again, variability, kind of almost like a U-shaped pattern here. But over these 12 years that the folks have been observing, we do see flowering occurring significantly later. And then again, we can't really say it's getting later without trying to get at why. And so the other thing that Erin did was compare these onset dates with the winter and spring average temperatures and total precipitation. And so this is the cottonwood flowering. So focusing in on this plot here now, just those three separate trees are indicated in the red, green, and the blue. Those are the different individuals. And then the black is the mean response. And what we're now doing here is plotting the winter precipitation total on the x-axis, and that is September through February rainfall, and then the day of the year on the y-axis. And in all cases, we see this significant relationship that's trending. It's a downhill slope. And so basically what that means is that with wetter winters, more rainfall in September to February, we have earlier flowering. And we can actually quantify that by looking at the slope of the line, the rise over the run, for each unit in greater precipitation, how many days earlier do the trees flower? And for every 10 millimeter increase in total precip in that period, the flowers advance about a day and a half. And then we can extend that same approach to the different species. So again, looking at New Mexican privet, we did the same thing. We just looked at, are there trends over time? We again start to see that kind of like funky U shape in some of the points. And in most of them, there's not a very strong pattern. But again, in flowering, there was. And I'm sorry, my red circle is covering over the confidence interval, but this was the same finding as with cottonwood, where the flowering in stretchberry also is showing a significant shift toward later flowering over this period. And it's going to look really similar, same plot for these guys too, where we've got the total 6-month winter precip on the x-axis and then the day of year on the y. Each of the lines is an individual tree. The black is the mean response. They're all doing the same thing. They're not going haywire here. But we have an even stronger trend. It's kind of hard to tell. The slope doesn't, excuse me, look that different, but it is. For the same amount of increase in rainfall, for every 10 millimeters greater rainfall during that period, we actually see those flowers leap forward by over 3 days. So same deal here. When you have wetter winters, these guys tend to flower earlier as well. And then let's look at leaf out in stretchberry. That's got that funky U-shaped situation here. Not a significant trend, but still a relationship with both winter temperature and cumulative precipitation. So this is an interaction here, and this is a little bit trickier to parse. And I have another plot that shows an interaction that is a little neater. But what we're seeing here is cumulative rainfall, again, on the x-axis. Again, the y-axis is the day of year for this privet. We're looking at leaf out, and we see that it leaves out when the winters are both warmer and wetter. And there's 2 factors that play a role here. For every 10 millimeter increase in that winter precip, the trees leaf out by, or the bushes leaf out by almost a day earlier. And for every 1 degree increase in temperature over that period, the leaf out advances 4 days. And so you can have an advancement because of both of those variables play a role, is basically what the take home is here. And then let's look at our other friend, the 4-winged saltbush, which was the shrub that didn't quite do what the other 2 did. We've got the same kind of situation here where there's a lot of scatter in when they put on their leaves or flowers, and when their flowers open and when their fruits are ripe. Not much of a trend in any of them, but we can look at whether there is a relationship with environmental variables and whether we tend to see earlier or later activity under particular conditions. And so with 4-winged saltbush, we see a relationship where the warmer winters, we look at mean winter temperature, that's December through February temperature, a huge advancement. Basically, for every 1 degree warmer we have in the winter, these shrubs flower almost 10 days earlier. So they are very winter temp sensitive, and that was across the board for all the plants again. Okay, let's look at trends in Eastern cottonwood. And I put this one in last because it's the interaction that's a little trickier to understand. This is where it looks like it's drifting towards getting later in the year over the period of record, but it's also kind of confusing and non-significant. And what we see is that these trees are both sensitive to winter moisture and winter temperature. And so what we've got here is the 6-month precipitation along the x-axis, and you'll see that when it's wetter, just like with the other guys, they tend to leaf out earlier. But there's also an additional effect of temperature. And so when it's cooler and wetter, you have a stronger effect of things advancing than when it's warmer and wetter. And I didn't put the interpretive values on that one, but the fact that the lines aren't parallel is what is indicating that both variables matter and are interacting. And so to kind of summarize this, and again, we just focused on those couple of shrubs and trees and just a couple of the phenophases. So there's so much more that could be done here. We can begin to kind of construct a picture that the species are sensitive and they don't respond in all the same ways, honestly, to the same amount of changes in variables, which is where you start to have stuff do different stuff in different years. So basically, just looking at the species and phenophases that we examined here, when we have 10 degrees more, or sorry, 10 millimeters more rainfall in the winter, you see the cottonwood flowers coming out about a day and a half earlier, but the stretchberry or the privet advances its flowering by twice that, and it leaves out a little bit earlier as well. And we see the cottonwoods leafing out earlier too. And then when we have a warmer winter, even by just 1 degree warmer, that privet leaves out 4 days earlier, but the four-winged saltbush is way ahead. It shifts almost 10 days earlier. And the cottonwoods are leafing out earlier too. And it kind of those, I didn't put the values on that because those two depend on each other. And so just kind of a quick little example of what this can lead to is if you have those kinds of situations where species are responding to different degrees, you can go from 1 year where you have, again, this is my really goofy cartoon of different species in flower and when they're in flower. You might have continuous nectar availability under one set of conditions, and then in another where things shift or contract or limit their flowering because of these kinds of shifts in the environment, you might have something like this where a gap opens up in the middle of the season, and then that's not as good. And so this is one of the kinds of ways that we're hoping to be able to continue to evaluate Nature's Notebook data going forward and be able to tell stories about how things are changing, what the consequences might be. Real quick, I wanted to just also demonstrate that in addition to taking that very focused view of the data that we just walked through for this time series that we have for this one location, the data also can be contextualized within all of the rest of the data that are being contributed to Nature's Notebook. And I have just a couple of very quick examples of where researchers have, rather than focused on one location, said, I'm going to wrap my arms around all the data that are available in Nature's Notebook and see what I can say. And this is an example where several years ago, researchers pulled all of the data contributed by Nature's Notebook, which are represented in the red triangles, and then combined it with records from herbaria, which are pressed plant samples where folks have documented where and when a plant was found, and we can extract information about whether it was in flower as well, and combine those different data sets to better understand what's going on. And what they did in this study was basically look at what are the environmental variables that seem to play the greatest role in what prompts plants to flower. And we already, as we've talked about in this presentation here, we already know temperature matters. That's what the Tmax, Tmin is. Mean annual temperature matters. Precipitation matters. But then what the cool take home from this study was was that there are also important variables that are often overlooked, which is things like the number of frost-free days or the beginning of when the frost-free period begins, or how much of the precipitation falls as snow. And so what the height of the bars here represent is how many species seem to be affected by or driven by all these different variables. And that's I don't really want to go too deep in that, just to illustrate that even if you're contributing data just at your one site, you never know how it might be used. It can be part of these very large scale studies that reveal very cool insights. And along those same lines, there's been a big increase in research lately looking at how the phenology of plants varies within urban areas and then less developed areas, partly because the more urban areas tend to be warmer, but also because there's a lot of artificial light in those areas compared to less developed equivalent environments. And so what we're beginning to really see pretty clearly is that there's an advanced start of the season and a delayed end of the season, leading to a longer growing season in areas with the additional warmth and light like we see in these urban environments. And so this is a relatively recent study where researchers had again grabbed up all the Nature's Notebook data that they could for, in the left, the breaking leaf buds, and on the right, the colored leaves for woody plants, and then layered it on top of this map that you can see that shows concentrations of artificial light. And they were able to show that indeed this pattern, as depicted here, is very much a thing. Things are extended by at least 6 days in either direction when you account for all the other variables like latitude and elevation. And this is, again, the kind of situation where just contributing your observations to the database, you never know what might happen, but these are really some pretty groundbreaking insights that are being gained. And so if you weren't familiar with that, I just wanted to point out that and almost any time a publication like the two that I just mentioned that come out in the literature, what we try to do is craft these publication summaries, which are just public friendly language that describes in our best attempt to tell the story of how the data were used and what was done there. And those are available on our website. And if you sign up for our newsletters, you'll be notified when these come out as well. And so kind of just to wrap up here, I want to really express gratitude to the Albuquerque BioPark team for their really incredible efforts and persistence over time. It's truly impressive and admirable what they've done here, the duration and the frequency of their monitoring. The consistent data that they have collected reveals fascinating insights and patterns, and I feel like we've just barely scratched the surface here. There's so much more opportunity, so much more. The data offer valuable local insights, and as I just kind of barely hinted at, really valuable when they're brought together with other observations too, like across the whole country. And I just want to take this opportunity to say that every observation matters, every data point matters. And so we are, even though we highlighted Albuquerque today, we're so grateful for everyone for everything always. And since I saw that there were so many folks on here that are new, in case you're just looking to learn more, there are all the good things going on with Phenology Week this week, and then later this week too on Thursday, Thursday or Friday. I think it's Thursday. I'm also going to be a guest on the Climate Central monthly climate brief, talking more about the spring index maps that I showed at the beginning, and just a little bit more about the NPN and phenology monitoring more broadly. So if you're interested in that, you're welcome to join that one too. It's open to the public and we can share this around after as well. And with that, I just want to say thank you so much, and I'm happy to answer questions if there are any.
So much, Theresa. It's amazing how much you can do with that data set, and there's still so much more to explore.
So much more.
I think there's a number of other local phenology programs and leaders on the call, and they're wondering, how can I have an analysis like this, or kind of create some of those cool bubble diagrams? So I don't know if you want to say anything about just the potential for like researchers' use of the data in general, or maybe some of our plans to keep digging into this year.
Yeah, it's such a fair question that I feel like I almost feel like I wish we would. We don't have a ready answer yet. That's the thing. Like we're still really in the middle of baking this cake. And so what you're seeing is kind of some of our first attempts at what are good ways to synthesize these data. Because the intention is to come up with, scripts and routines that either we can work with you to run, or we can even hand the tools over the kind of to give you the keys to drive the car kind of thing. We're trying to figure out the best way to accommodate that because everybody has different skill sets and interests in either driving or having it driven for them. Um, but we are very aware of the need for this, and we really want to be able to meet you where you're at for achieving those kinds of insights. And so, yeah, I guess on one end of the spectrum, the data are freely available. Anybody can access them and download them. And if you're so inclined, kind of go it alone. They're available on the website through a download tool called the POP, the Phenology Observation Protocol or Portal. We also have an R script called RNPN if you're savvy like that, or there's the visualization tool, which you can do some stuff in, but not quite the plots that I showed today. But then that's kind of the deep end of the pool, and then kind of walking back out to the shallower end. That's the part that we're still really actively trying to work on and develop. And I don't know if I want to put any time constraints on promises. Erin, what would you say?
Yeah, I think it's something we still need to explore. I think we're really interested in trying to create more kinds of dashboards and things for local phenology programs. So that is something we're going to be looking into later this year. And we will have an opportunity for having a working group of local phenology leaders to help us think through that. So yeah, stay tuned for that later this year. Let's see. I think we're a little bit over time, but we can maybe. One other question was about, what are species that are maybe most important to look at? Maybe that is applicable for not just the local phenology programs, but also our independent observers. And in the chat, we put a link to the campaigns, but I don't know, Teresa, if you want to say anything else about just what species might be most useful.
I always come at it from what's most useful to us is what keeps you engaged. Because we don't have a crystal ball, and that's the hard part. We don't have an overarching single question that we're aiming to answer here. If we did, it would be much easier for us to say exactly which species to monitor, where, when, how frequently, so that we could know we were getting exactly the data we need. But because we're constructing a data resource that can be used in ways that nobody has even imagined yet, as well as a million ways that have been imagined, you can't parameterize that. And so what matters is to have the consistency in observations regularly throughout the season and optimally over multiple years. And so for that, I really turn it back on what would make you happy and keep you engaged.
I think that's a great answer. I did drop the link to the campaigns again. Yeah, you could try to find something on that list, but I think that's a great suggestion to find something that's convenient and that interests you. And we'd love to have you collect data with us for multiple years. Well, I think that we'll wrap it up at this point. Thanks so much, Teresa, for that great presentation. Thanks to all of you for being with us on our first day of Phenology Week. I hope that you'll join us on social media as well this week. We're using the Phenology Week hashtag, so feel free to send in photos and other things that you're seeing this week. And then we'll hope to see you on some webinars the rest of the week as well. So thanks,
Thank you.
everybody.
Thanks, Erin.
Bye.
Bye-bye.